Let's get started. Good morning, good afternoon, good evening to all of you that are joining from all across the world. I'm Florent Junot from the Singapore Immunology Network in A-STAR, and today it's my pleasure and honor to host and introduce our global IMMUNOTALK speaker, Professor Chris Glass. But before doing that, a reminder that we will have Professor Gwendolyn Randolph next week on September 8th, so stay tuned. And please do join us again on next week on September 8th. So today we have Professor Chris Glass from University of California, San Diego, UCSD, presenting live today from the West Coast. So Chris Glass is a physician scientist in UCSD, USA, where he is a distinguished professor of cellular and molecular medicine, and also a distinguished professor of medicine. And you know, as we say as usual, we like to share like the origin of our speaker. Chris acknowledged to be a native Californian living in San Diego. So a bit of history. Dr. Glass majored in biophysics at UC Berkeley and received MD and PhD degrees from UC San Diego. He performed internship and residency training in internal medicine at Brigham and Women's Hospital in Boston before returning to UCSD for fellowship training in endocrinology and metabolism. Dr. Glass was a founding member of the Department of Cellular and Molecular Medicine at UCSD. So Chris Glass is recognized for his work on molecular mechanisms that control macrophage function in health and disease. His laboratory has had a long-standing interest in elucidating the molecular mechanism by which sequence-specific transferrin factors, co-activators, and co-repressors regulate the development and function of macrophages. A major focus has been on understanding the roles of nuclear hormone receptors and other signal-dependent transferrin factors in controlling program of gene expression that underlie both their normal homeostatic function in healthy tissue, as well as their pathogenic function in the arterial wall, adipose tissue, and brain. So, as we'll probably learn today, current studies in Dr. Glass' laboratory use a combination of genetics and genomics to define general molecular mechanisms that establish macrophage identity and cell-specific response to homeostatic and pathogenic signal in mouse model and human subject. His laboratory is applying this approach to understand and modify pathological programs of macrophage gene expression that promote the development of atherosclerosis, diabetes, cancer, and neurodegenerative disease. So, of course, Chris has received multiple prestigious awards and honors for the accomplishment, and has been elected to the American Academy of Art and Science, the National Academy of Medicine, and the National Academy of Science. So again, Chris, thanks so much for accepting our invitation and for giving us this today, the Global IMMUNOTALK, entitled Decoding Macrophage Phenotypes in Health and Disease. But just before you start, we have a question for you about your career. And the question today is, could you name one thing you would similarly, or one thing you would do differently if you were to start your scientific career again? Chris.
Well, Florent, thank you for that wonderful introduction, and to you and Alina and Carla for the opportunity to present at this great seminar series. I would say this is of the things that are positive and negative about this last year of COVID. This is definitely on the positive side, and so it's really a pleasure to be part of it. So, yeah, things that I would do again. You know, you mentioned I came through an MD PhD program. My original idea was to be a physician. But in medical school, I was seduced by the excitement of medical research, and so I ended up, you know, combining those and following a research career. But I would say the medical school experience, I like to think of as the ultimate survey course on life, and it really gave me a very broad background, and I would definitely do that again. You know, things that I would do differently, I would have paid more attention to my statistics classes as I was going through school, and being able to really, you know, deal with complex data sets more rigorously. So that would be my advice to the up and coming, you know, students: pay attention to your statistics class. Okay.
So I think thanks. I'm also regretting probably this part also. So thanks for sharing this with us. And please, Chris, you can start to share your slides, and the floor is yours for the talk.
Okay, great. Thank you, Florent. And I'm going to apologize. I'm getting over a cold. One of my nieces brought her kids to the home 2 weeks ago, and the little kids are definitely vectors. And I picked up something that's not COVID, but might as well be. I was pretty down and out for a while. Anyway, so what I'd like to do is to tell you about where we are with trying to develop a sort of a general approach to decoding macrophage and microglia phenotypes that takes advantage of our understanding of enhancers. So just, to put this into perspective, I think, this is an immunology seminar series. Everyone's familiar with macrophages as key players in the response to infection and injury, initially discovered by Metchnikoff. And we know now that they play these critical roles in innate immunity through their ability to recognize pathogens and pathogen-associated molecular patterns, their ability to phagocytose and to make a broad range of signaling molecules that regulate the functions of other cell types. So what's become of great interest, I think, over the last decade or so has been an appreciation of the roles of macrophages beyond immunity and their functions in tissue homeostasis. And so we've learned that during development, tissues are populated by macrophages, and each macrophage develops its own phenotype in part through instructive signals of the tissue it resides in. So microglia would be an example where specialized roles include synapse refinement, and Kupffer cells would be an example of cells of macrophages that play roles in iron homeostasis. Now, our interest in macrophages also are very much focused on their roles in human disease. When I started my lab many years ago, it was really focusing on macrophage foam cells and atherosclerosis. And over the years, we've studied macrophages in a variety of disease contexts that include roles of tissue macrophages in insulin resistance, in neurodegenerative disease, in cancer, and so on. And these studies have really raised a number of questions as to why and how macrophages that are really there for adaptive purposes use their various functions in ways that ultimately contribute to tissue pathology. And so our approach to trying to understand this is really to try and dig into the mechanisms that regulate macrophage gene expression. So what I'm going to do is to just initially give you an overview of our current working model for transcriptional regulation of macrophage identity and function. This is what we call a collaborative hierarchical model, and then use this as a sort of a starting point for understanding how tissue-specific macrophage identities are established. And here I'll describe some recent work on identification of environment-specific signals that drive the selection of Kupffer cell-specific enhancers. And then finally, address how these identities are altered in disease. And there discuss reprogramming of the myeloid cell populations in a diet-induced model of NASH. And then give you some of our thoughts on these recently identified macrophage phenotypes referred to as SAMs, DAMs, and LAMs. So the major focus of the lab is to study enhancer promoter interactions that are used to establish cell-specific and signal-dependent gene expression. And so what you're seeing here on the left is sort of a general model indicating that the promoters are the obligatory start sites for messenger RNA, but these regions don't have enough sequence information to enable genes to be expressed in their appropriate developmental time frame, or in the context of cell-specific gene expression, or how they respond to signals. And so this additional information is largely provided by enhancers. And one of the key aspects of enhancers is that these turn out to be the major sites in the genome that are bound and activated by what we would refer to as lineage-determining transcription factors, or the combinations of factors that are necessary to establish a particular cell fate. And so through these interactions of cell-specific enhancers and promoter elements, then one achieves appropriate signal-specific and cell-specific gene expression. Now, a key aspect of the talk today is to take advantage of the ability to quantitatively measure various aspects of enhancer and promoter function. And so we can do this using a variety of assays. One is referred to as a TAC-seq, and this is an assay that measures open chromatin. An example of this data is shown here on the right in the vicinity of the BIN1 gene, and these peaks indicate open chromatin at the BIN1 promoter and at upstream regions about 10 kilobases away. Now, open chromatin is an indication that transcription factors have bound and that they've shifted the position of nucleosomes, but it's not really a direct measure of enhancer activity or promoter activity. And to gain some insight into this, we use chip sequencing for other marks, one being histone H3 lysine 27 that's been acetylated. This is highly correlated with promoter and enhancer activity, and here you can see ChIP-seq for H3K27 at the promoter, but also associated with these open regions of chromatin upstream. So this to us is telling us that these regions are likely to be active enhancers. And then to try and understand the basis for their selection and activity, we can perform ChIP-seq for transcription factors, and here's an example of a chip for PU1, a key macrophage lineage-determining factor, and you can see it's bound at the open regions of chromatin here at the promoter and also upstream. And then finally, it's often the case that enhancers don't necessarily regulate the nearest gene, and so it's often very valuable to perform some sort of an assay for chromatin confirmation. And so one assay that we frequently use is PLAC-seq, and these loops indicate that these upstream regions are in close proximity to the BIN1 promoter, suggested that they actually do play roles as enhancers for this gene. Now, a major question that we've been pursuing for many years now is, what are the combinations of factors that drive the selection and function of enhancers in macrophages in various different contexts. And the working model that we developed now more than 10 years ago, and which for the most part is standing the test of time, is what we call a collaborative hierarchical model. And the idea is that there are a relatively small set of lineage determining factors that play major roles in driving enhancer selection. And here I'm illustrating two of them, PU1 and CBP. And these proteins, when they are co-expressed, are able to bind to regions of the genome that contain the recognition motifs when they're relatively close together, within about 100 base pairs or so. And so this interaction we refer to as collaborative, because PU1 can only do this when CBP is present, and CBP can only do this when PU1 is present. And mutations in either one of these sites will also prevent the binding of both factors. So this collaborative interaction results in what we refer to as a primed enhancer. It establishes open regions of chromatin that can then be accessed by other classes of transcription factors. And one example that we've studied extensively is NFKB. So if macrophages are activated with LPS, NFKB moves into the nucleus, and a major site of action is at these primed enhancer elements, which then allows the recruitment of co-activators and enhancer activity. And to give you a real world view of this, here's a gene in macrophages, prostaglandin E synthase, that's expressed at low levels in resting macrophages, but is highly induced in response to TLR4 ligation. And these top two tracks are illustrating the binding of PU1 and CBP in the resting and TLR treated condition, and you can see they're pretty similar. So they're co-binding, and there's a collection of binding sites here about 10 kilobases upstream where both are bound. But in the absence of treatment, these regions are not occupied by NF-kappa-B. However, when the macrophage is activated, we see NF-kappa-B entering the nucleus and binding to these exact locations that were set up by the combination of macrophage lineage determining factors, and this leads to enhancer activity. So really a key point here is that we get a cell-specific output of a broadly expressed signal-dependent transcription factor because it's basically been told where to go in the genome by the lineage-determining factors for that cell type. Now, if we carry out genome-wide motif enrichment of all of these peaks that gain acetylation in response to the TLR4 agonist, what we find is the most enriched sequence is a motif for NF-kappa-B, followed by motifs for PU1 and CBP. Now this definitely fits within the paradigm of TLR4 signaling, which leads to the activation of AP1, NF-kappa-B, and IRF factor. But it actually adds quite a bit of additional information. So this model, which is a generic model for how these receptors signal in multiple cell types, does not take into account the cell specificity of the response. And the motif analysis does tell us something about that the ability of NF-kappa-B to go to these places is dependent on P1 and CBP. And the other point, which I think is interesting to think about, is that what we're doing is we're recovering motifs from hundreds or thousands of enhancers. And this motif enrichment is telling us what the most important motifs are. And so from this analysis, we can say that even though all of these factors are being activated at this particular point in time, what the enhancers are telling us is that the NF-kappa-B site is the most important site in driving gene expression. So the enhancers are essentially giving us a sort of a data reduction approach to help us understand what I've often used the analogy of what the cell's thinking. And so what we want to do here, and this is really the main point of the rest of the talk, is how can we exploit this? So we can measure enhancer activity in a quantitative way, and we know that the transitions in enhancer activity states are driven by sequence-specific transcription factors. So what we can try and do now is use this property as a way of inferring factors and upstream signaling pathways and receptors and environmental signals when we don't have any idea what's going on at all. And so the idea is that if we can carry out this analysis before and after a transitional event that leads to changes in hundreds or thousands of enhancers, the motif analysis will give us the identities of the factors that played major roles in driving this transition in cell fate or cell state. And so now I'm going to give you an example of that. And this is an effort to try and identify signaling pathways and transcription factors that drive the selection of Kupffer cells. So these are specialized macrophages that live in the liver. They're of embryonic origin. They reside on the sinusoidal endothelium. And for us, one of the reasons that we study them is that they're among the largest populations of tissue-resident macrophages. So we could get a lot of them for our genomic assays. But they're very interesting. They participate in a number of important physiologic processes, such as iron homeostasis. They're sensors of gut-derived microbial products. They're important players in the liver immune system. and they play roles in the initiation and resolution of inflammatory diseases. And so this project, which I'm just going to describe very briefly, was led by Machido Sakai, Ty Troutman, and Jason Seidman. And it was based on Machido developing a very specific CRE driver in which he introduced CRE and TD tomato behind the CLEC4F gene, which others, including Martin Julian, had shown is very Kupffer cell specific. And then he bred this mice to a ROSA26 lock-stop-lox diphtheria toxin receptor mouse. And by giving diphtheria toxin, he could very rapidly deplete Kupffer cells. And what he found, and this is very consistent with Martin Julian's work, is that very quickly, the empty niche is populated by recruited monocytes, which then over a period of approximately 2 weeks become very similar to the original embryonically derived Kupffer cells. And this is just a heat map showing the changes in gene expression of the monocyte-derived cells and their convergence to the Kupffer cells. Now, one of the striking findings in this study was that very rapidly after the monocytes had entered a depleted Kupffer cell niche and formed a contact with the sinusoidal endothelial cells, transcription factors that had been considered to be lineage-determining transcription factors for Kupffer cells were very rapidly activated. And so this is most clearly seen for NR1H3, which encodes the liver X receptor alpha. That is highly induced within 12 hours of the monocyte touching the depleted sinusoidal endothelial cell niche. And we see other factors that are also implicated in Kupffer cell state fate transitions, such as ID3 and SPIC coming up very quickly. And this is then followed by upregulation of the more classical Kupffer cell specific genes like CLEC4F. Now, some genes really take a long time or don't get turned on. TIM4 is an example. And other genes, which are high in monocytes, are rapidly downregulated. And so this was the, sort of for us, a test case for, can we figure out what's going on here by studying enhancers? And so what we learned was that this initial phase of rapid activation of Kupffer cell lineage determining factors is based on the activation of a set of poised enhancers that are in the circulating monocyte. And so this is shown as an example in the vicinity of the LXR alpha gene. There are these regions of open chromatin in the monocyte that are open at the beginning and stay open. And these regions show very rapid induction of H3K27 very rapidly after the cells enter. So this goes along with the rapid activation of LXR. Whereas in contrast, the chromatin environment in the vicinity of the CLEC4F gene is really basically closed in the monocyte. And this doesn't really come on. We see a little hint here at 48 hours, but it really doesn't come on until a week or so. And so that is consistent with this process being the selection of new enhancers, presumably through collaborative interactions. And so if we carry out motif analysis of these peaks, we get many motifs that correspond to what we think are Kupffer cell lineage determining factors. And I'll just focus on these three, the liver X receptor, RBPJ, and SMAD2/3, and just in a cartoon tell you what Machido, Ty, and Jason found through analyzing these enhancers over time. And so RBPJ was one of the motifs that was enriched in enhancers in the monocyte. This is a downstream target of the Notch pathway. And what's very interesting is that in the absence of Notch signaling, this is a repressor. So it maintains a open region of chromatin in a repressed state through the recruitment of a co-repressor complex. In response to Notch signaling, the intracellular domain is cleaved, moves to the nucleus, displaces the co-repressor, recruits a co-activator. And so now what you have is a switch in the enhancer from being a repressed enhancer to an activated enhancer. And this is what we think is turning on a significant number of the lineage-determining factors such as LXRa, SPICE, and others. And so once these factors are turned on, they can then participate in this in a version of this collaborative model that I've showed you. Now we have new Kupffer Cell specific lineage determining factors that can interact with the generic macrophage lineage determining factors like P1 to set up the Kupffer Cell specific enhancers. And so this series of studies, which led to the identification of Notch signaling and then roles of BMPs, came out of an enhancer analysis and was actually very concurrent with and in agreement with studies from Martin Gillian's lab, who used a more of a bioinformatics and computational approach. So we arrived at the same conclusions through different pathways, and that I think is one of the other sort of sources of reassurance that this general approach may be valuable. And so then I'd like to now, for the rest of the talk, consider how these identities are altered in diseases, and again, stick with the liver and tell you about our studies of reprogramming of myeloid cell populations in a diet-induced model of NASH. And so this is a very common condition. There's actually a spectrum of what is referred to as non-alcoholic fatty liver disease. It tends to be associated with various risk factors, include obesity, diabetes, and hyperlipidemia. And the initial phase is referred to as simply non-alcoholic fatty liver disease. It's characterized by deposition of fat in hepatocytes and liver enlargement. This condition is largely benign, but it can progress due to inflammation and the initiation of fibrosis. And among this set of individuals, a subset will go on to develop frank cirrhosis, in which there's hepatocyte necrosis, fibrous septa formation, and ultimately liver failure. And then a subset of these patients can go on to develop hepatocellular carcinoma. Now we can model this in the mouse using a variety of methods. And for the studies that I'm going to show you, we used a diet model that induces steatosis. So these are these large white vacuoles, inflammation. So this liver is full of additional mutant cells that aren't normally present, and fibrosis. So it's indicated here by picoserous red staining. And this work was led by Jason Seidman and Ty Troutman, and what they started with was a single-cell RNA-seq experiment of the CD45 positive immune cells in the liver. And I think most of you are probably familiar with these types of t-SNE plots. What you're looking at is basically the cells that were captured in the analysis. Each dot is a cell, and they're clustered according to how similar they are to each other. And in this left t-SNE plot, the cells are color-coded according to whether they came from the liver of an animal fed the chow diet or the NASH diet. And this was probably the most striking single-cell experiment that we performed, because there's almost no overlap in the cells that we recovered from the wild type or the NASH diet fed animals. Now we can put names onto these cells based on their patterns of gene expression, and this big cloud here in the middle is predominantly a cloud of either the original Kupffer cells that we called KC1 cells, or Kupffer cells in the NASH liver, or recruited Kupffer cells. And so if we quantify these, in the healthy liver, what we're calling KC1 cells they're by far the dominant CD45 positive cell in the liver, accounting for about 90% of the cells. But after, in this case, 30 weeks of this NASH diet, this normal population of cells is basically gone. And it is replaced by what we're calling KC2 cells, which are derived from KC1 cells, but also a monocyte-derived population that lives in the same niche that we're calling Kupffer niche recruited macrophages. And then there are other myeloid cells that are much more represented than they are in the healthy liver. And so we carried out a number of experiments that included lineage tracing and microscopy. And this was greatly aided by Anita Gola, who was a graduate student in Ron Germain's lab. And just to summarize what all of this, how we interpreted all this data is as follows. And that is, in the healthy liver, we mostly have normal Kupffer cells, and after the NASH diet, they transition to a disease-associated phenotype that we're calling KC2 cells. Now, these cells gradually die and are replaced by blood monocytes that recognize the empty niche and undergo a convergent pattern of gene expression. And so we're calling these Kupffer niche recruited macrophages. And then other blood monocytes take on different trajectories based on the micro niches in which they ultimately end up in. And these, I think, are very interesting and important for disease, but I'm really, for the purposes of this lecture, going to discuss the KC2 and KNRM cells. So what Ty and Jason were able to do, based on the single cell analysis, was identify markers that allowed them to purify each of the cell types that they had identified by single cell analysis. Somebody's working right next to me, and if this gets to be too loud, I'm going to have to leave and go out and tell them to stop. I wasn't anticipating construction happening during the middle of the talk, so we'll see what happens. Okay, so this transition of Kupffer cells from normal to the NASH phenotype is associated with changes in about 800 genes. So that's why they don't cluster in the same place on the TISTI plot. And these cells, the Kupffer niche recruited macrophages, even though they have a very different developmental origin and haven't been in the niche as long, they are very similar to the NASH original Kupffer cells, having less than 100 genes different. Now, what is going to be a major theme for the rest of the talk is that both of these genes turn on a gene called, both of these cells turn on a gene called TREM2 to very high levels, as well as CD9, and other genes that are associated with the upregulation of this pair of genes in other disease-associated macrophage populations. And this was interesting to us in the context of the Kupffer cell, because Ramachandran and colleagues, as this work was being done, published a very extensive single-cell analysis of human liver non-parenchymal cells. And there's a lot in this paper, but one of the key findings was the dramatic increase in a population of macrophages that they called scar-associated macrophages. And these were characterized as macrophages that resided on the fibrotic scars within the cirrhotic liver. And what they found was that these cells showed a dramatic increase in TREM2 and CD9, and they called these scar-associated macrophages. And so this discovery could be put into the context of prior work on disease-associated macrophages, and these have been looked at in various ways, but I'm highlighting some of the more prominent paper. So Ito Amit's laboratory in 2017 described what they call disease-associated microglia in the context of Alzheimer's disease, and then subsequently defined a lipid-associated macrophage population as being these macrophages that surround apoptotic and necrotic adipocytes. So these cells have been known for a long time, but what Ito and his colleagues showed is that these are a TREM2 high expressing cell. Then I've just told you about the scar-associated macrophages in cirrhosis. And so in each case, these are macrophage subsets for which the discovery was really significantly aided by single cell analysis. And that's really illustrated nicely here in the Amit paper from 2017, looking at microglia in a mouse model of amyloid that is a widely used model of Alzheimer's disease. And so this blob of points here represent the microglia, and it's just this little sort of pimple here at the bottom that corresponds to the cells that have the DAM phenotype and that have upregulated TREM2. And this was of interest because it had already been known that TREM2 plays an important role, a protective role in Alzheimer's disease based on loss of function studies in the mouse from Marco Colonna's work and others. And what's generally observed in these models and actually in humans that have TREM2 mutations is that in the absence of TREM2, the microglia have difficulty in migrating to the sources of plaques, and this leads to an expansion of the plaque. And so the interpretation is that this is a protective function of TREM2, and why loss of function leads to increased risk of Alzheimer's disease. Now, I'll point out that in this study from the Amit laboratory, the development of the DAM phenotype was suggested to occur through a TREM2 independent phase that transitioned to a TREM2 dependent phase. And so that's important. I'll come back to that later. And as I mentioned, they had also described this LAM phenotype. So L stands for lipid. So these are lipid-associated macrophages. They have a high lipid content. And when mice are deleted, when TREM2 is deleted from the mouse, interestingly, this phenotype of large adipocytes actually becomes much worse. Adipocyte area goes up, and the mice exhibit worsened metabolic parameters, such as worsened glucose tolerance tests. So putting all of this together in terms of common features of DAMs, LAMs, and SAMs, they tend to be associated with focal areas of tissue pathology, and I think of them as being in areas where there are phagocytic challenges. They appear to play protective roles, at least in the brain and adipose tissue. They're partially dependent on TREM2. That's been established for SAMs and DAMs by knockouts. All of these cells contain intracellular lipid droplets. If you compare the three cell types, there actually aren't that many genes that are overlapping in all of them, and that's I think that's interesting because each of the tissue environments is different. The common genes include CD9 and some others shown here. One thing that I think is important to point out is these don't really line up very well with M1 or M2 macrophages in any way. And so to get to the very last part of the talk, what I'd like to do is sort of serve this up as an example where there are some very significant knowledge gaps where this concept of using dynamic enhancers may be helpful. So this is a figure from one of Ito Amit's recent reviews on TREM2. And so TREM2 is a cell surface receptor. It has many ligands, and so this is one of the challenges of trying to figure out what it's actually engaged with in a pathological tissue context. But it's known to signal by forming association with DAP10 or DAP12, leading to the activation of SYK and PI3 kinase. But there are some very significant gaps in knowledge. So what are the signals, receptors, transduction pathways, and so on that turn TREM2 on? And what are the signal transduction pathways and factors that are downstream of SYK and PI3 kinase that mediate TREM2 specific functions? And so now I'll come back to these Kupffer cells and their transition from a healthy Kupffer cell to a sick Kupffer cell. And this transition for us was a really interesting opportunity to study what happened to a tissue-resident macrophage as the tissue went from healthy to sick. Because we knew that these cells came from these cells through lineage tracing experiments. And so Jason and Ty were able to collect enough of these cells to carry out quantitative measurements of open chromatin with the ataxi and H3K27 acetylation by ChIP-seq. And here we're looking at the TREM2 gene, which goes up, and a Kupffer cell specific gene, CD163, that goes down. And what you can see is that there's open chromatin at the TREM2 promoter and intronic region that has fairly low levels of histone acetylation, and that in the response to the diet, we now see a new region of open chromatin appear downstream of the TREM2 gene, and very significant increases in H3K27, both upstream and downstream. So this, to us, we would call that a NASH-induced enhancer. And then conversely, the enhancer landscape for CD163 basically collapses. Now, we can look at this at a genome-wide level, and this is a big effect. So when we looked at enhancers based on H3K27 activity, we saw that the NASH diet resulted in an increase in more than 4,000 at more than 4,000 sites by at least a factor of 2, and a loss of enhancer activity at about 3,500 sites. And when we carry out motif analysis, we see very strong enrichment in the enhancers that gain activity for AP1, NFAT, RUNX, and EGR motifs. Now, interestingly, at sites that are lost, we see a loss of the LXR binding sites. So, and this goes along with basically a loss of many of the enhancers that are Kupffer cell specific. We can line up these motifs with factors, and in fact, we find that EGR2 and ATF3 are highly induced, very likely contributing to these motif enrichments. But the LXR really doesn't change, despite the fact that we're losing motifs at Kupffer cell specific enhancers. And so what Jason and Ty did to try and get further insight into this was to actually carry out chip sequencing for LXR in the healthy and NASH liver. And what they found was they lost a lot of Kupffer cell LXR binding sites at Kupffer cell specific enhancers, but they actually gained almost an equivalent number that were associated with NASH specific enhancers. And when we carried out a motif enrichment analysis, this set of new LXR binding sites was highly co-enriched for ATF3. And so Jason and Ty carried out a chip for ATF3, and what they found was that LXR and ATF3 were co-bound at more than half of the NASH gained enhancers. So this is really a dramatic reprogramming of LXR binding and function in response to the diet. And so here's an example of this. LXR basically disappearing from a Kupffer cell specific gene and then being recruited to the TREM2 locus, including this NASH specific enhancer in what we think is a collaborative binding event with ATF3. And so putting all of this together, you know, what we're looking at is the conversion of a healthy Kupffer cell to a NASH Kupffer cell that has some characteristics of the LAM and SAM phenotype. These cells are gradually dying and being replaced by monocyte-derived cells that exhibit convergent gene expression. And at the molecular level, what we're seeing is that I didn't have time to show you this, but it basically at the standard lipid homeostatic genes like ABCA1 and ABCG1, which LXR regulates in all macrophages, that program didn't change. But what we observed is that at sites of Kupffer cell identity genes, where LXR would bind with other Kupffer cell lineage-determining factors, in the healthy Kupffer cell, that was lost in the sick Kupffer cell. And instead, LXR was reprogrammed to bind to NASH-specific enhancers through the upregulation and collaboration with ATF3. And so in terms of our general conclusions, we think this provides good evidence that one can use dynamic enhancer landscape to achieve insights into transcription factors and upstream signaling pathways that establish tissue and disease-specific macrophage phenotypes. And one good example of this would be the SAMs, DAMs, and LAMs. These are recently identified disease-associated macrophage phenotypes with overlapping but distinct gene expression profiles, and I would say as yet poorly understand functions. And so what I've shown you is, at least in the the SAM phenotype of Kupffer cells in a mouse model of NASH, we think that ATF3, EGR2, and LXRs are implicated in this cell state conversion. And we're now in the process of looking at mice in which we've deleted these genes and are putting them in the context of the NASH diet. So with that, I'd like to again recognize the major contributions of Jason Seidman, Ty Troutman, and Yoshida Sakai. They were assisted by a number of people in the laboratory, and we also enjoyed a great collaboration with Anita Gola and Ron Germain. And with that, I'll stop, and I'd be happy to take questions if there's time.
Thank you very much, Chris. Really a tour de force. Sadly, there is in the IMMUNOTALK format, there is no questions. However, it's a good transition for me to explain how you can get questions. The question and answer will be via Twitter for people which are interested to reach out to Chris. As it is explained on this slide, you can search for account Global IMMUNOTALKs. You can find a tweet that says ask a question for Dr. Christopher Blas here, and reply to that tweet with your question and mention at Global Immuno. So with this, thanks again, Chris, and thank you very much. And please ask on Twitter a question for Chris. See you next week. Bye bye.